You may be knowing oxidative phosphorylation, the process of ATP synthesis that happens due to the oxidation reduction process of the enzymes, coenzymes and cytochromes coming across the electron transport system inside the mitochondria. And the process is called by different names like cellular respiration, respiratory chain, electron transport system, respiratory chain, internal respiration, electron transport chain, ATP synthesis. Now here to give an abstract of how the raw material for the process of oxidative phosphorylation appears. You say the coenzymes NAD and F-AD are used during the metabolic activity of carbohydrates, proteins and fat metabolism. Even you may be familiar with the words glycolysis, oxidative decarboxylation, Krebs cycle, beta oxidation and deamination of amino acids into the keto acids, which may possibly get into Krebs cycle. So that way, there the NAD and F-AD acting as the coenzyme at different steps will be reduced into NADH and FADH2. And in turn, this NADH and FADH2 will be oxidized. And the electrons due to this oxidation process will be released into the respiratory chain and the electrons will be passing through these redox reactions where the oxygen will get reduced at the end and then the ATP synthase will be activated by the proton pumping, which will be resulting in the formation of synthesis of ATP inside the mitochondria. And here just we have given an overall idea about the total amount of energy formed out of the metabolism of glucose. You may be knowing C6H12O6 that stands for the glucose, and by the involvement of oxygen, glucose will be metabolized into carbon dioxide and water and the resulting energy. Now here about 686,000 calories of energy released from the metabolism of 1 mole of glucose. Now this is the amount of molecules present with 1 mole of glucose, and this will be the total amount of energy out of 1 mole of glucose, and that occurs spontaneously in the mitochondria. Now here these are the regions where the process of oxidative phosphorylation may occur. That is, the oxidation of NADH and FADH2 will occur and the electrons will be passing to the final stage of the formation of ATP synthesis. Now here you see the mitochondria with outer membrane, inner membrane and then the matrix inside. Now the inner membrane having the infoldings into the matrix and the surface of the inner membrane provided with a stocked F1 particle. Now the space between outer membrane and inner membrane is called intermembrane space or perimitochondrial space, and then the matrix, then the F1 particle will be carrying the ATP synthase enzyme, and then the inner surface of the inner membrane and outer surface of the inner membrane would be active in the process of oxidative phosphorylation. So there happens these two processes of the transfer of electrons as well as the transfer of protons, which will be enabling the reduction of oxygen and then the formation of ATP, that we will explain in detail with the diagram. Now here these are the enzyme systems, coenzyme, enzyme, cytochrome systems involving in the process of the oxidative phosphorylation. Now the malate shuttle, glyceraldehyde 3-phosphate shuttle. Now they are important in transferring NADH available in cytoplasm into the mitochondria. Just for an idea, during glycolysis, which happens in cytoplasm, NADH is produced, and the NADH may not, now it's said the mitochondrial membrane is impermeable to NADH. So the electrons available with the NADH transferred to the inner part of mitochondria by the malate shuttle, and that way. But anyway, the remaining metabolic activities like oxidative decarboxylation, that is pyruvic acid to acetyl coenzyme A, and then the Krebs cycle, and even the beta oxidation of fatty acids, all may happen within mitochondria using the mitochondrial NAD and reducing it into NADH, and that would be available inside the mitochondria. And now here the malate shuttle is very much useful for transferring the electrons with the reduced NADH into the mitochondria because of the impermeability of mitochondrial membrane to NADH. So that is the first step in taking out the electrons with the NADH under the enzyme coenzyme complex called complex 1. And then succinate dehydrogenase, succinate ubiquinone reductase, helpful for oxidizing FADH2 into FAD. That way the electrons available with this FADH2 will be utilized and will be taken into ubiquinone, that is coenzyme Q, available at the oxidation reduction process, mediating the electron from this NADH and FADH2 to the cytochromes. Now here come the cytochromes like BC1 and then A3. These are all the cytochromes involving in the transferring of electrons to the final part of the electron transport chain. Now to go in detail with this diagram illustration, here you see the NADH is oxidized to NAD by the NADH dehydrogenase, which is also a part of complex 1. That way, even you assume that the electrons coming from the cytosol region to this part by this malate shuttle system. Anyway, the electrons available which will be moving towards the ubiquinone, that is coenzyme Q. Now during the movement of this electron, there the production of energy which will be enabling the H plus protons available in this matrix region into the intermembrane space through the inner membrane. So the energy that's produced during the movement of the electron sufficient for the pumping of hydrogen from matrix region to inner membrane space at the oxidation of NADH to NAD by the complex 1 enzyme system. That way you can mean. Now here we have abbreviated this intermembrane space and inner membrane as well as this matrix by IMS. And then this FADH2 will be oxidized into FAD, as we already told, in the presence of complex 2 involving that succinate dehydrogenase, and the electrons received out of this will also be getting into this coenzyme Q. That way the coenzyme Q is reduced by accepting the electrons out of the NADH and FADH2 when they are oxidized into NAD plus and FAD respectively. But anyway, the coenzyme Q may transport 2 electrons at a time, and the electrons going through this Q will be taken into the cytochrome C, which will include both these cytochromes B and C1. Now these electrons passing from Q to C will be reducing the B cytochrome, which will be having the heme as a part, as its part, non-protein part, or otherwise you mean it like a prosthetic group. Now it would be existing in the form of Fe3 plus, that is oxidized state, on the receipt of electron, which will get reduced to the ferrous state Fe2 plus, and then it will be settling, the electron will be settling to C1, which also already in the form of Fe3 plus, yes, here it should come 3, and which will be oxidized to reduced to Fe2 plus. That way that the electron will be settling through this B cytochrome and C cytochrome, and the energy produced during the movement of the electron in addition to oxidizing and reducing the B and C1 cytochrome, enabling the pumping of protons from the matrix region to intermembrane space. And there it is said the membrane, mitochondrial membrane is permeable to the freely available hydrogen protons. That way you can assume the intermembrane space is occupied with a more amount of protons, hydrogen protons. That way there is produced an electrochemical gradient which may be pumping the hydrogen protons from this intermembrane space towards the matrix. And the process of the migration of the hydrogen protons from intermembrane space towards the matrix due to such osmotic gradient called a chemiosmosis, which will be stimulating the ATP synthase available with the F1 particle and helping the conversion of ADP and inorganic phosphate into ATP. Now the energy that's produced during the proton pumping may be useful for the formation of ATP, in which the energy is captured, by which we say this ATP bond is filled with energy on one side due to the proton pumping from intermembrane space to the matrix region. And then this electron, after coming out of these cytochrome redox reactions, will get into one more cytochrome A3 complex, that is complex 4, which will be accepted by the oxygen, which is said to be the final electron acceptor. That way the oxygen will combine with the hydrogen protons to form H2O. So the oxygen is acting as the terminal electron acceptor, So on one side there the electrons are accepted and will be getting into the formation of water. On the other side, the proton pumping will be enabling the formation of ATP, which captures the energy that's produced during the movement of proton from intermembrane space to matrix region by the process called chemiosmosis. Now here you can assume the passage of electron from the step of oxidation of NADH and then to the formation of H2O by alternative oxidation reduction process of all these steps as redox reaction, and then the energy produced out of such movement said to be redox potential. So that way the ATP is synthesized with the energy produced by the proton pumping and then the movement of electrons.